Hello everyone, this video is about Chapter 1 Biodiversity, Subtopic 1.1 Biodiversity and Classification. This is the learning outcome for this lesson. At the end of the lesson, students should be able to state types of biodiversity, genetic, species, and ecosystem, and state the hierarchical classification. State types of biodiversity, genetic, species, and ecosystem. Biodiversity is defined as the variety of living organisms based on genetic diversity, species diversity, and ecosystem diversity. Biodiversity can be divided into three types, which are genetic diversity, species diversity, and ecosystem diversity. Let's take a look at each of these biodiversity. Genetic diversity is defined as genetic variety within a species. Thus, it is considered as the total number of different alleles of genes within a species. New genetic variations can arise by gene and chromosome mutation, and also by sexual reproduction, where new combination of existing genes occur through crossing over and independent assortment. Species diversity is defined as variety of species present in a community. Ecosystem diversity is defined as the variety of ecosystem present in the biosphere. Biosphere refers to the entire portion of the earth inhabited by organisms. State hierarchical classification. Hierarchical classification is a system of grouping things according to a hierarchy or levels and orders. Organisms can also be classified based on hierarchy from the highest level of classification or less general classification, which is domain, to the lowest level of classification or very specific classification, which is species. Hierarchical classification of organism is based on 8 taxons. The taxons are domain, kingdom, phylum, class, order, family, genus, and species. This is an example of hierarchical classification for cheetah. The domain is Eukarya. The kingdom is Animalia. The phylum is Chordata. The class is Mammalia. The order is Carnivora. The family is Felidae. The genus is Panthera. And the species is Panthera pardus. The scientific name for cheetah is Panthera pardus. Taxonomy is the science of naming and classifying organisms. Scientific name and classification system is based on binomial nomenclature. This system is developed by Carolus Linnaeus. The two-part scientific name of a species is called binomial. The first part of the name is the genus. The second part is species. The first letter of the genus is capitalized, and the entire species name is italic or underlined for handwriting. This is an example on how to write a scientific name. Escherichia coli is a type of bacteria. Escherichia refers to the genus. The first letter, which is E, is capitalized. Coli refers to the species. The letter C is in small letter. When handwritten, Escherichia coli is underlined with 2 separate lines. When typed, Escherichia coli is italic. Escherichia coli can also be written as E. coli or Escherichia species. Note that in Escherichia species, only Escherichia is underlined or italic. Subtopic 1.2 Domain Bacteria and Domain Archaea. This is the learning outcome for this lesson. At the end of the lesson, students should be able to state the two domains of prokaryotes, bacteria, and archaea, and state the importance of bacteria. State the two domains of prokaryotes, bacteria, and archaea. Let's review back the general structure of prokaryotes. Prokaryotes mostly have plasmid, mostly have capsule, they have cell wall made up of peptidoglycan or murin. They have 70S ribosome, they have villi, they are unicellular and sometimes also found in colonies. They have diameter in the range of 0.5 to 5 micrometer. They have flagella. They are single circular DNA molecule with no histone associated, and they have DNA not enclosed by a nuclear membrane. Last but not least, they have no membrane-bound organelle. Prokaryotes can be divided into two domains, which are bacteria and archaea. The first domain is bacteria. For example, E. coli. E. coli has no nuclear envelope surrounding the bacterial chromosome. Thus, it has no true nucleus. All of the genes required for basic survival and reproduction are found in the single chromosome. Some E. coli cells also contain plasmid, which is a small DNA molecule in a circular form that carry genes for certain specialized function, including resistance to specific drugs. The second domain of prokaryote is archaea. An example is Sulfolobus species. Sulfolobus species grow in volcanic springs with optimal growth occurring at pH 2 to 3 and temperatures of 75 to 80 degrees Celsius, making them acidophiles and thermophiles respectively. Sulfolobus species cells are irregularly shaped and have flagella. These are the differences between domain bacteria and archaea. The cell wall of bacteria consists of unique layer of peptidoglycan, while the cell wall of archaea consists of polysaccharides and proteins. In domain bacteria, DNA is single circular molecule and not associated with histone protein, while in domain archaea, DNA is associated with histone protein. In domain bacteria, the lipids in the plasma membrane are composed of unbranched hydrocarbon chains connected by ester linkage, while in domain archaea, the lipids in the plasma membrane are composed of branched hydrocarbon chains connected by ether linkage. State the importance of bacteria. Bacteria is important in recycling the chemical elements in ecosystem. Some bacteria can act as decomposer and nitrogen fixer to return the chemical elements back into the ecosystem. Examples of these bacteria are Rhizobium species, Nitromonas species, Nitrococcus species, and Nitrobacter species. Bacteria also plays a symbiotic role. For example, enterobacteria like E. coli in human intestine. E. coli live symbiotically in the human intestine of human aid in digestion of food materials that human intestine cannot break, for example lactose, and breaking cellulose in herbivores. Some bacteria also pathogenic, meaning they can cause disease. Example of pathogenic bacteria is Salmonella species. Pathogenic bacteria can cause typhoid fever, food poisoning, and gastroenteritis. Salmonella poisoning is often linked to contaminated water or food, especially meat, poultry, and eggs. Bacteria also important in research and technology. Bacterial plasmid is used in recombinant DNA technology for the production of human insulin. This is the end for this lesson. See you in the next video.